So my name is Rajesh Ranganathan. I was introduced in the other room. I'm the director of the Office of Translational Research at NINDS. I've been on the job since January, so I'm new to the role, new to NIH as well. I've only been at the NIH a year and a half. And prior to that, I had background in pharmaceutical company, Novartis, where I had a variety of roles I won't go into at this point. And so my job here is to chair this session where we have several speakers, including Steve, come join us, who have sort of small presentations that they have prepared to sort of address certain programs that they wish to highlight. But the focus, what we're hoping is to spend a better part of the hour really sort of addressing questions you might have. And that's what we wanted to leave enough time for. So in my opening remarks, I was going to just make a few comments about the programs we have in the Office of Translational Research that are directed towards therapy development, if you're not aware of them. And I won't be able to go into any detail unless you specifically want to know more about them directed by questions. So there are 5 sort of main programs within the office. So the 2 that I just mentioned that I'll be speaking about, the anticonvulsant screening program and the counteract program. So the anticonvulsant screening program is a program, a long-standing program that NINDS has had to help develop therapies targeted against epilepsy. And it's run as a contract with the University of Utah, where compounds submitted by a variety of investigators in academia or industry get triaged through a series of in vivo models, and the data then gets shared back with the supplier for furthering their drug development enterprise. And the program has been widely respected in the field and has been successful in having a hand, some major, some minor, in several anti-epileptics that are in the marketplace and in clinical care. And the program, and I will specifically be addressing on next week some of the changes we're implementing in the program as we try to sort of strengthen the core of what already exists and moving into some new vistas that have been recommended by review groups that have looked at the program in greater depth. That's all I'll say about that program. In terms of the second program, Counteract, which I'll talk about next week, so by the way, the first one is a sort of a $5 million a year investment from NINDS's perspective. The second program is a Counteract program, which stands for Counter Agents Against Chemical Threats. And so this is a sort of a post-9/11 program that has a broad umbrella of biodefense against biological threats, radionuclide threats, and chemical threats. And NINDS is responsible for the last one, which is the chemical threat piece of it, in a trans-NIH manner. So we're responsible for the program for all of NIH. And the reason for that is most of the chemical threats tend to be nerve agents that lead to epileptic fits, and many others similar other neurological problems that we try to address by developing antidotes that the government could then stockpile to use in the case of an emergency. But there are lots of spin-off effects on what that program is doing that actually has relevance to other diseases in neurodegeneration and places where neuroprotection might actually be generally relevant. The 3 other programs in the portfolio are the Blueprint Neurotherapeutics program, which is a new program, about a year and a little bit older than a year, where the NIH is experimenting whether we can behave like a virtual pharma, wherein we have contracts with multiple contract research organizations to do the various steps in the therapy development process, starting from medicinal chemistry all the way through to taking things into early proof of concept trials in humans. And investigators, both from companies and academia, again, can apply to the program, and they don't necessarily get a grant in the usual sense of the NIH funding scheme, but they get services in kind. So essentially, stuff gets done at these CROs, and the data shared with them. The IP remains with the PI throughout that process. So that's an experiment we're doing that is of interest, and there's an upcoming final receipt date for that program in October that we've received letters of intent from numerous companies and academics that we're interested in seeing apply to the program. The SBIR SDTR program, I won't say anything more because you'll hear more from Stephanie about that specifically, which also sits in the Office of Translational Research, and that's another sort of $40 million investment because it's a set percentage of the NINDS budget. So I spoke to you about the anticonvulsant screening program, the translational program that Stephanie will talk about, Blueprint Neurotherapeutics, and Counteract. The only other program left to mention is the cooperative program in translational research that has two components, an exploratory component and then a development component. And this is another $40 million investment on the part of NINDS to support projects that essentially are trying to walk this realm across therapy development. And here, unlike the Blueprint one that I just mentioned, which is focused on small molecules only, this one and these programs support devices, small molecules, antibodies, and have a very broad mandate. And you'll also hear from Kip in the context of programs that are really focused on devices and technology and how they'll move forward. Also on the panel today is John McHugh and Steve Groft. John's from NCATS, which Story mentioned, and a specific program called TRND, Therapeutics in Rare and Neglected Diseases. And he'll talk a little bit about that program and how it actually interfaces specifically for this audience with advocacy groups and how it's done that. And Steve has been the head of the Office of Rare Disease Research for quite a while now, and he'll give his perspective on how to think about partnering on diseases that are rare by definition, but how advocacy groups can certainly help bring more research dollars to bear, but also perhaps bring synergies to move the research forward there. I wanted to give a couple of examples of nonprofit group development that I've learned about in the 7 months I've been in the job, which have been kind of interesting and maybe sort of exemplars that you might take some lessons away from. The first one is in the cooperative program, which is the internal lingo is it's a UO1 program, which was started a while back in Batten's disease, which Story also mentioned. It has achieved an investigational new drug with the FDA and has gone on to clinical trials. But the preliminary data were obtained through a partnership with two advocacy groups that were really focused on essentially pushing that effort forward. And so that's a way where early work gets funded in terms of seed money that Bob mentioned that can move something forward to then having robust funding from NINDS to move all the way into clinic. We've also received, for example, an application in spinal muscular atrophy where there's a gene therapy project where the application includes a partnership with an advocacy group where they're committing companion funding to cover portions of the work that were not requested in the actual application that we'll end up funding. So that'll support overall the objectives of taking that thing all the way to therapy development. And the Huntington's Disease Fund initiative, Cure Huntington's Disease group has essentially set aside money and asked for NINDS to essentially use our peer review process to pick things in the Huntington space for target validation. So they have sort of, as Bob indicated, they've tried to essentially leverage the NIH mechanism to say, you guys do this well, we don't have to replicate the entire process internally, you do this, but we'll give you the money that is set aside essentially for this particular purpose. So these are just examples of ways you might think about partnering, and there might be others that we can discuss in the open forum. So I'll just end with just a last sort of comment about, I think there are three ways I can think of at least that nonprofits can certainly help in therapy development space, which is what I end up thinking about on a daily basis, which is we need investigators to know about the funding mechanisms that exist and so that they can apply to the most effective mechanisms. So to the extent that you become familiar with the entry criteria for the mechanisms and can direct investigators, both academic and companies, to us, that would be very, very advantageous for them, but also for us to essentially have a robust program. The second is where you're able to, and I know that different groups have different abilities to do this, and where you're able to co-fund projects to essentially move things forward or fund ancillary studies that are not core to therapeutic development, but are really important at the end of the day for it being adopted by a larger entity downstream, is also another way to think about helping. And I think this is an important thing that we're going to focus on in our office in the coming months and years, is how do we effectively transfer things that we fund to the next funding body, whoever they might be downstream, whether it be VCs, venture capital, or large companies, or other, or even certain nonprofits could also take them on. Is that we don't necessarily, at the moment we achieve that IND in a preclinical space, but then they're sort of left to fend for themselves, if you will. And I'd like to see us play a more active role in essentially sort of being a matchmaker for the next step. And I think, again, a partnership with advocacy groups here could be very effective because you have I would imagine a complementary Rolodex to things that we have that would allow these entities to open doors for these entities. So I'll stop there and switch over to essentially Stephanie to give you an overview of the SBIR. I was going to introduce each speaker, but I'm not going to do that in the interest of time because otherwise we'll just not actually cover any of the material and have a discussion. So if you want to know more about Stephanie, ask her later.
So the small business programs are commonly referred to as the SBIR and STTR. It's what a lot of people know them as. It stands for the Small Business Innovation Research Program or Small Business Technology Transfer Program. And these are a set of programs that are congressionally mandated set-asides for small businesses to do the research and development needed to bring something towards commercialization. So right now, the combined set-aside of the SBIR and STTR is 2.95% of the NINDS's extramural research budget. That's about $42 million in fiscal year 2012. Now, these programs have a relatively broad scope. They can support everything from therapeutics to diagnostics to development of research tools for neuroscientists. In addition, we can support things all along the development pipeline, from in vivo, in vitro work, development of a device prototype, all the way through to clinical trials. And in fact, NINDS does support clinical trials, both phase 1 and phase 2 clinical trials through this program, as well as translational research, that later stage translational research that Rajesh was talking about. Right. So the other important thing to note about these programs are that they will be changing over the next year. And this is due to the recent reauthorization of both of the programs. Over 20 provisions in the reauthorization will be directly affecting NIH. Now, I'm not going to go over all 20 provisions today, but I do want to touch on a couple that you might find interesting. So first, in the area of eligibility, the venture capital participation will be changing. So historically, companies that were majority owned by venture capital firms were not eligible for the small business program. Soon, not yet, but soon, some of our SBIR funds will be able to go to those companies that are majority owned by multiple venture capital, hedge, or private equity firms. Now, as I said, this isn't taking place quite yet. If you do have companies or individuals who would fall under this provision, who would be interested in the small business program, have them contact me and I can give them the implementation timeline. But this will be coming. The other thing I want to talk about are the budget award sizes. So while in the reauthorization, the general guidelines for the size of the award haven't really changed very much, and you can see they're up here, the flexibility associated with those award sizes will be changing. So historically, NINDS and NIH as a whole was able to award applications above those general SBIR STTR guidelines, provided there was adequate justification for that project. That is not going to be the case in the coming year. We will be capped, as per the reauthorization, to 50% over the guidelines that you see here. Now, there's language in the reauthorization indicating that the Small Business Administration, and the Small Business Administration sets all the rules for the small business program, the Small Business Administration will be able to give us a waiver in specific instances for specific projects or programs, but they've indicated to us verbally that that's going to be a relatively rare occurrence. So the vast majority of our applications are now going to have to come in under this cap. So information about the budget and actually general information about the reauthorization can be found in the policy directives for the specific SBIR and STTR programs. So everything that doesn't have to do with eligibility is found in those policy directives. And they're currently open and available for public comment. So for those of you who are very familiar with the program and would like to look and see the changes that are coming down the pike and would like to comment, I would encourage you to do so. The other big thing that the reauthorization did is increase the set-asides over the next 6 years of the reauthorization. So we have 5 years left. Today is this year, 2012, is the first year of the reauthorization. So overall, our set-aside of the extramural research budget will be increasing. So in the interest of time, I'm going to kind of conclude with some challenges and what I see as potential opportunities. So our set-aside will be increasing, but the size of our awards will be getting smaller. So we will be funding more awards in this area. And we're very interested in solid, strong, high-quality applications so we can keep a high-quality set of programs going. So if you know of small businesses that are working in your disease area, please have them give me a call. I'd be happy to talk to them about the program. The other thing is that we would like to increase commercialization, and we think that we're going to need to do that through increased partnerships. As these size of the wards are getting smaller, companies are going to need to seek either other funding sources or other partners to help take the product the rest of the way to the market so they can help those patients be used by clinicians and be used by researchers. That means that they're going to have to develop partnerships with either investors, whether it's venture capital firms, angel investors, pharmaceutical companies, or even nonprofits like yourself. And with that, I'm going to conclude. I put the NIH SBIR website up here if you can't see it. It's sbir.nih.gov. It has information about the programs in general, as well as information about the reauthorization, including a rough implementation timeline. I also have my email address up here. If you have any questions about the small business programs, feel free to contact me. I'm happy to talk about them.
So Kip is one of those excellent but overworked program directors that Bob talked about in the Division of Extramural Research. So,
all right. So there's a simple way to introduce myself. I'm the device guy. If you have questions about devices, you come to me. And there's kind of a sad misconception about engineering devices. Engineering devices is all about the ability to communicate. And unfortunately, when you picture an engineer, you picture Revenge of the Nerds or the cast of The Big Bang Theory. You don't picture Brad Pitt or George Clooney. There's a reason for that, and that's where nonprofit organizations are critical in helping what is probably the most iterative process towards therapies. Now, I have listed on this slide 3 areas in which the NIH contributes to developing devices. The number one and the most commonly overlooked is that devices and technologies help perform basic research. They can make it cheaper, they can make it faster, they can make it more accurate, and they can open new areas of research. Right now, if we're studying function of the brain for Parkinson's, we stick 100 probes in the brain, which, not surprisingly, can change the function of the brain. If you want to create a better technology, if you want to really influence therapies down the road, you need to get in at an early stage. Secondly, we fund the initial development of lab prototypes for neuromodulations, strategies for, again, Parkinson's, for spinal cord injury, etc. And this is where often there's not enough collaboration at an early stage. Engineering and engineering devices is inherently designing for a consumer, an end user, which is a patient or a clinician. And right now, often, there's not an input at an early stage over not what is a statistically significant improvement, but something that's usable to a patient, something that makes a functional quality of life improvement. And that has to be an iterative process, even more so than potentially drug development. And finally, translating devices from lab prototypes to robust clinical use. A lot of what the NIH funds is either small businesses or academics who have some familiarity with creating these lab prototypes, but they don't have the familiarity with getting through the regulatory process. They need to be partnered with larger institutions that have experience taking a medical device, which can have thousands of parts, software, firmware, etc., that all have very specific regulatory pathways to get through to the clinic. If your Windows desktop freezes up, you reboot your computer. If your pacemaker freezes up because the software doesn't work, it's a completely different level. So there are many opportunities for nonprofits to get in at this early stage. First of all, because engineers aren't particularly good at communicating, there needs to be a facilitatory role in getting engineers to talk to basic scientists. There's engineers who can do hundreds of different things, and there's people doing the science who don't know they can do it. And it's just a simple matchmaking process that needs to be done. And we do that at the NIH level, but we need help. Secondly, defining a market. For these to be viable products that really help basic research, you really need to be able to identify a market. And a lot of times, an engineer has an idea, but really doesn't know if they can sell it and make it a viable process so they don't proceed forward with it. And then finally, of course, we've talked a little bit about the SBIR program and more limited funding. A lot of these things cost a lot of money, and a lot of the numbers that we're talking about isn't enough to fund at the level that needs to be done to do something important. This is where it becomes even more important at the proof of concept level, is actually talking to bioengineers and neuroscientists to actually define what are meaningful quality of life improvements to the patient. It's called design inputs in engineering speak. We need you to say what you need, and then we will tell you what we can provide. And it has to be this iterative process, because those aren't always going to match. But unfortunately, at an early stage, this is often overlooked, and you have an engineer saying, this will be a cool improvement, and then especially for rehabilitation assistive devices, you often find something going to market that just patients won't use. And finally, in terms of, and this is pretty much obvious to most of the people in the room, I suspect, when you're actually translating to the clinic, it becomes even more difficult for a basic research scientist who have never translated before to actually deal with all the peculiarities of the regulatory pathway, including enrollment, talking with clinicians. This isn't just talking with one group. This is actually facilitating communication among the people who will be implementing the device, implanting the device, monitoring the device over time, and the patient population, making sure it's not something that just works in the clinic, but works for the patient in the home.
Thank you. So John also comes with a pharma background. He was at Wyeth for many years. He's a medicinal chemist by training. And as I said, he's at NCATS, and he'll tell you more about TRND and Bridges, another program that you might.
Thank you for the opportunity to talk. I'm going to try to be concise, because every time I talk in front of Steve, I take up all of his time, and I want to make sure he has the opportunity to talk about all the wonderful things the Office of Rare Disease Research is doing, and to give you all opportunities to answer some questions. So there are two programs, as Rajesh said, that I want to talk about: Therapeutics for Rare and Neglected Diseases and the Bridges program. And I'm going in inverse order. We're going to talk about the Bridges program first. So both of these programs are programs, resource programs for preclinical development. So particularly the Bridges program is a late preclinical development program. And the model really is a collaboration between a staff that I have in my branch at the NIH that's experienced in all levels of drug discovery and development, from early assay development all the way through to toxicology, and clinicians who, through a solicitation, go out and find projects to work on with outside collaborators. They come to us with, in this case, a clinical candidate. They apply to work with the program, and we go through a fairly extensive peer review and due diligence process to select projects that we want to work on. And then we move forward to do a gap analysis and look at the equity that's brought to us and decide what studies do we need to do to enable our collaborator to file an IND. And that's really our goal. We take in all types of modalities: small molecules, peptides, oligos, gene therapy, antibodies, recombinant proteins. We take the whole gamut. And really our goal then is just to make sure that we enable our collaborator with a package that they can file an IND on. And this is strictly a resource program. We don't provide any funding. We provide expertise, and we provide data that's very valuable to our collaborators. And the eligible applicants can be any academic, both in the U.S. or outside the U.S. We have had a number of nonprofit folks be partners in the applications that go forward, as well as other intramural government labs and SBIR eligible businesses. So right now, that's part of our charter, that if you are a small business, you have to be SBIR eligible. So this little twist about 25% of the funding being available for VC-funded companies, we haven't yet figured out what we'll do for our next solicitation, because that's the only time it'll be relevant going forward. But currently, that's the requirement. So the last on the bottom is the website that will take you to our new Bridges website. Just a little snapshot on intellectual property, because that's a very key part, particularly for small businesses that are in this area. In a case like this, where you're taking in a clinical candidate, most of the projects that come to us have defined composition of matter or intellectual property around them already. So there isn't often a problem of trying to figure out how to deal with intellectual property developed jointly. I wanted to highlight just the rare and neglected these are all just the rare disease projects that have come into Bridges over the last 7 years. So we have a total of 30 approved projects, and 10 of these, so about a third of the portfolio, were in rare neglected disease projects. And then 5 of these we've completed all of our work, and then our collaborators have advanced these through to IND filing, early clinical development. And I would like to highlight, particularly because Ron is here, Ron Bartek from the Friedreich's Ataxia Research Alliance. This was one of the first applications we brought in, and it was at a time where we required a advocacy group, an academic, and a company. So we had very high hurdles, and Ron's organization was able to pull all those pieces together, come to us with a piece of equity, and we provided the data to enable him to file his IND with his collaborators. And as you know, the project has progressed tremendously through phase 2B study. I think Ron has been able to manage a changing group of collaborators to continue to advance this project. But this is a key example of, I think, how we like to work and reach out to the nonprofit community, the patient advocacy community. This is a very successful example of how bringing the right people together with the right resources from the NIH can really advance research and therapeutic development in an area that's really really near and dear to your hearts. And the other projects, again, are against a variety of orphan designatable targets, and have advanced quite well just with the academic collaborators who brought them in. So I'll shift gears just for a minute and talk about the TRND program. The Bridges program anybody can apply to, and I just wanted to highlight the 30% of the applications we've gotten in rare disease space. But the TRND program, you have to come to us with a rare or a neglected tropical disease as your starting point. The model is similar. It's a collaboration between the intramural group that we have built up and extramural labs with disease area and target expertise and the project to come in. In this case, the TRND program will allow you to enter much earlier. So in a small molecule setting, as early as what I would call early lead optimization. And we have medicinal chemists on hand, as I said, biologists. We will continue to develop your chemotypes. We will advance them. We will do toxicology testing, pharmacokinetics, whatever it takes to move your molecule through IND filing and into early clinical development. Our goal for both of these programs, but particularly for the TRND program, is to take what may be a risky asset and may be unable for you to go out and get external funding and provide you with a data package that de-risks that molecule. Our goal is not to take this molecule through to registration studies in the clinic, but merely to give you a data package that will enable you to go out and get external funding to finish the development of your molecule so that we both get what we want, which is new treatments for patients. Again, for TRND, we have a variety of different modalities. We'll accept peptides, small molecules, antibodies, enzyme replacement therapy. We don't do gene therapy yet for TRND, but most other things are available. And again, academics, nonprofits, government labs, small business, but also in this case, we don't have an SBIR eligible criteria to come in, so we've worked with big companies, small biotech companies, virtual biotech companies, and we don't have a requirement for the applicants being in the US. So it's fairly wide open. We have a solicitation that is open right now. It will close September 30th. We'd love to increase the number of applications that we're getting. The website at the bottom will give you a list of application instructions on how to apply online. But the TRND program is a little bit different in terms of intellectual property. If you come to us with an early lead molecule and we do all medicinal chemistry to optimize it, that's a place where we can actually jointly develop intellectual property. So we have a number of agreements, such as a CRADA, that would govern those agreements and also enable us to determine what to do with the jointly held IP. This is our current portfolio, and my apologies to Ed in the back. His company decided to change their name, and I haven't updated my slide yet. You'll have to tell me why you chose an ancient Phoenician city to name your company, Sarepta. We have a number of projects across a broad range of rare disease targets, many of them in the NINDS space, neurological space, but also in many others as well. And I just wanted to highlight 3 where there's very strong advocacy involvement and continuing involvement. One of those is with the Niemann Pick-C project. This is a project which began when a patient advocacy organization, the Ara Parsegian Medical Research Foundation, came to us and asked us to do a repurposing screen to find a drug in a phenotypic assay that might impact that disease. We've built an entire project around it, multiple investigators, academic. Right now, we have Johnson & Johnson involved. We have a different advocacy group, the NPC SOAR advocacy group, that's involved in this. And we spent 2 hours yesterday going over with this patient advocate our plan for phase 1 and later trials. So it's a case where long and continuing involvement with the patient advocacy group helped us decide what are the questions that the patient community needed to have answered, and which molecules did we have the ability to use to answer those questions. The third project, or the second project that's highlighted here, is a project in CLL that we're doing in collaboration with the Leukemia Lymphoma Society and Kansas University's Institute for Advancing Medical Innovation. Again, these are three parties that have been together since the very beginning, and LLS has played a really key role in, again, defining what the patient needs are, connecting us through their large network of research collaborators, as well as provided funding to enable us to move this molecule through an ongoing phase 1 clinical trial that's been going on for about a year. The last one I want to highlight is our other Duchenne muscular dystrophy project. It's with a company called Revirgin Biopharma, which is an interesting company because it's solely funded using venture philanthropy from muscular dystrophy associations. And we've partnered with them, and we've actually partnered with the MD associations to decide how best to handle the two funding streams that are coming in, and dividing up the plan that we're going to use to develop this molecule through the clinic. So I think that highlights three different ways that advocacy groups have worked with us on these projects to help advance therapeutics development. Once, the first one, they helped us start the project and help us define how to go forward. In the second one, it was connecting resources and patients together with our development. In the third one, it's almost co-funding of a company that's dedicated to developing therapeutic for Duchenne muscular dystrophy. So with that, I will stop, and Steve still has a little bit of time, so I'm happy. Here's all the contact information, and if you do want to find out more about the trend solicitation, that's the best email to send something to.
Okay, so the last short talk will be by Steve, and this challenge will be to keep it short, I guess.
Thank you, Rajesh. Ready to go? Okay, great. Thank you, and thank you for coming to the forum that NINDS has put together. It is really a great opportunity to learn who your program people are, and to work with them and to understand the programs. I think that's really to me, one of the most significant parts is you get to know the resources that are available to you. And in my slides later on, we'll talk about this. I just want to mention David Eckstein in the back of the room. I'm be cutting out, not quite in the far back, but he'll be here the rest of the day, or a good part of the day. So if you have any questions about the office, direct them to David, and we'll get them. But you can always contact our office and me, and we'll discuss issues, whatever you want to focus on. I'm going to talk today a little bit about the movement from the traditional task of patient advocacy groups to where we are today, and what you're going to be looking at as a patient advocacy group and a leader, because we are looking at evolving roles, and changes are coming. And if you haven't moved forward, you're going to be called in to ask and to answer to your constituents, what are we doing and how are we going to do this? And they all require resources and a lot of work. That's one thing we're finding. So here are some of the traditional tasks that many of you have confronted and worked through and are now operating. Establishing your medical and scientific advisory board, supporting research and training programs. How often you're requested to provide some bridge funding or some additional funding for these programs. You're contacted by the media to give information about your disorder, about the research that's ongoing. You are definitely the main link towards the patients and the physicians often. So you have many responsibilities. Then the difficult problem oftentimes when results come out from research, you're called upon to translate those to your constituents, your patients, your families. What does all this mean? You're asked to help organize research conferences, and you do this with your family conference. The main time you do it in conjunction with the research conferences. So there are a lot of aspects that you're asked to do. And part of this is really as you go on and with your own patients, you start to identify the differences in the patients. Well, what's really going on here? And so many times the differences in how the disease presents itself really are reflected in some genetic differences, and you're the ones who often will observe these because you see hundreds, if not thousands, of patients and say, gee, some patients are a little bit different. We ask you to join advisory councils and special committees here at NIH to help advise us on what the best course and direction frequently will be. You're often asked to participate in institutional review boards as members of the public to help give advice. So where are we now with the evolving roles? And each one of these, as the last slide showed, it takes a lot of energy and time and effort to really to fulfill these needs. The way things are coming up now, we're looking more and more at a globalization of research, so it's going to require you to identify your global partners in the patient advocacy community and the research community as you move forward. You'll hear some more things about patient registries and developing a curator or gatekeeper to that information. Tremendous amount of effort is underway here at the NIH and in the community to develop these registries as a source of information to begin to understand the disease a little bit better. You also will be asked to help develop biospecimen repositories and collect samples from patients. Again, a major need for the research community. And now, rather than being a member of the institutional review board, you're going to be the sponsor. You're going to be the one who will have to go out and find an IRB to look at several of your studies. Whole different role than what you were in before. We also are asking you to help identify patients to participate in both natural history studies and clinical trials, because it is a major need as we go along. And I will say, the success, as was mentioned about Ron Bartek and so many of the others, the success of the trial really does depend on the ability to recruit patients to participate. If we don't have the patients, the studies, the research is not going to move forward. And so the last one that John referred to and talked about extensively was really to expand the knowledge of translational research resources and opportunities that are available from many of the institutes here at the NIH. And you're going to get a very nice snapshot of what's going on here within NINDS that can be useful. There are resources, and a lot of this really will be determined by how well you're able to effect collaboration and coordination. A major role. I hope this comes up all right. You see some of the coordinated efforts, and all of the parties around the wheel there with the spokes going out, you probably will be speaking with and talking with and strategizing how you're going to get greater emphasis on your disease and research. And so we're finding more and more the patient advocacy groups and their scientific medical advisory boards are the ones who are forming the steering committees that will actually get things to work. And it's just it's a different approach, but there are some good models available from different patient groups, and this is a little bit different way of showing the various partners who you're going to have to work with coming along, moving forward as far as developing potential products for your disorder. So there are a lot of partners, but there are good models, there are good examples, and there are people who are willing to help identify who to work with and how to work with them. The Institute folks, we in the office can also do this. So I'm going to end it with just two little comments, and I included these slides mostly for your information, and I won't talk about them, but the Patient Registry Initiative and a lot of other activities. I do want to just get to the next slide. And this, there was a question about the scientific conferences program. On our website, we have got probably all of our conferences, the 1,100 or so conferences that we support with the institutes. We have an agenda, the participants, and a brief summary, and even links out to publications. So if you're looking for a previous conference and you can't find it anywhere, please come into our website. It should be located there. If it isn't, give us a call. We've got actually hard files on everything related to previous conferences. And I do want to just mention too that the Rare Disease Clinical Research Network that you see mentioned there, and currently there are 18 consortia. I believe 7 of them are with the supported by the NINDS with our office. We'll be recompeting this. Probably the announcement will be next year, something to look at and to become familiar with. It's a great opportunity to begin to move research in your disease, and we think it's a pretty good research network, and I think you'll become familiar, if not already, with NeuroNext and many other opportunities here at the Institute. So I will end it with that, and if we have time for questions, be okay, Rajesh?
Oh, thank you. So floor is open. I guess the one comment that I failed to make in my introductions that I wanted to sort of maybe come back to is sort of when I think about translation and I think about sort of therapy development, one of the things, at least from my own pharma experience and others may have experienced similarly, which is that projects tend to go awry when they haven't thought about the patient. Projects tend to go awry when they haven't thought about who this is actually intended for. And therefore, it's actually very important to think about that as early as possible. From a very practical perspective, in terms of thinking about, what you're trying to develop, is that going to actually be helpful to the population? And are you developing it in a way that's going to be helpful? Perhaps in a case you're developing something that's an injectable that'll never be used because it's the wrong thing to be using in that particular setting, and you need something that's oral, or vice versa. And in other situations, it actually also has a practical impact in terms of whether it's actually going to be commercializable at the end. If you haven't thought through that process, you might have the best project that goes all the way but then falls flat because it doesn't actually have the traction to make a difference in the clinical setting. So in that sense, your engagement with that community of patients is extremely critical to actually provide that input right up front. I sort of think of translation, often gets depicted even on my slide, I'm guilty of that as well, as a chevron diagram that has a unidirectional component to it, but it's not really true. There's a feedback loop to it, and it's actually maybe a virtuous circle where we need to think about what it is that the patient is telling us about what we need to do in research and translation to help them. So with that, the floor is open. Please use the microphone, otherwise it doesn't show up on the video.
Thank you so much. So many questions, and I see other people have their hands up. So why don't I start with just two very simple questions. One is for Dr. Roth. The last 3 slides you showed was a treasure trove of information. How do we get our hands on those last 3 slides?
Well, they'll be made available over. We're going to do it.
There's a fee.
Okay. And then
called user fees. No, we're not going. No, we'll send them to you.
Okay, great. And Ms. Fertig, I mean, it's very exciting, the reauthorization and the the opportunities it creates. You didn't mention a timetable, or if you did, I missed it.
So there's an implementation timeline that's up on the web. Briefly, things are going to start being implemented the next calendar year. So the eligibility guidelines, the final eligibility guidelines will come out at the end of this calendar year, and they won't go into place until that occurs. And then for those of you who don't know, our general omnibus solicitation for the SBIR program, it's kind of the overall NIH solicitation for SBIRs and STTRs, gets put out in January of every year. So some of those changes will start being implemented then, and some will be earlier. So if you have any questions, look at the sbir.nih.gov website. It's a treasure trove of information as well. But if you have any other questions, feel free to contact me.
Sharon?
Okay, so this is another question for Stephanie. You always hear a little bit of background grumbling about the quality of the SBIR projects that get submitted. Do you have any sense, is there any specific way they tend to go wrong, or is, in general? And I'm asking because a lot of times, I've even been asked to read applications for companies because I think they struggle with this idea of writing a grant. And is there anything consistent that you see that would be good advice for them?
So I think I guess it depends. I will say the fact that the companies are asking you to read those grants, that's great. I think one of the things that a lot of companies do struggle with is grantsmanship issues. Many of them have never come to the NIH before. Some of them may be their principal investigator came from industry, so they've never had experience with the NIH process. And I think that could be hugely helpful to help guide those companies. I think that, some of it has to do with, we do have a congressionally mandated set aside. We are required to spend that money on small businesses. So it's not as if we can say, well, we didn't get applications this year, so that means we can put the money somewhere else. It must be spent on the small business program. And so that means if we're not getting applications in, that enough applications in to make up that set aside, well, then there you go.
So if I can just maybe add one comment to that, which is, I mean, this is coming into this job. It's one of the things I've sort of tried to shine a light on to say what can we do differently to actually increase the quality of what's coming in, and also sort of the partnering on the other side as well. And one aspect to that, the current the change of sort of having VC funded companies coming in. If you just think about it, by default, if people are going to VCs as the first choice of funding, you're automatically looking at the lower quartile, half of the bucket, so to speak, and that's now changes the dynamic. And so I would hope that actually fundamentally changes that, with the caveat that obviously Stephanie mentioned, that the caps don't really help us. It sort of is a fighting force on the other direction, because it's now giving us even little less money to do the substantive work with, and so we'll have to figure out how to deal with that.
So I'm Melanie Leitner from Prize for Life, which is an ALS nonprofit, and our model is pretty different than I think a lot of patient advocacy organizations, where one of our big focuses is on infrastructure, tools and resources. And so my question for Rajesh is, as a nonprofit that's focusing, we're not as interested in a single company coming through, or a single academic, or a single approach, but really building things that can be useful across multiple academics, multiple companies, specifically targeting translation. So I didn't hear anything in your presentation about opportunities related to these sort of infrastructure issues around biomarkers, patient stratification, databases, data sets. And so can you say a little bit more about your evolving thinking on where, obviously these things are critical towards meeting the translational gaps, but it's not clear to me how to interface with NIH around any of those issues.
So yes, you're highlighting a gap in the portfolio as it stands at the moment. And in fact, at the moment, I've been sort of championing this idea of essentially providing funding to develop, especially pharmacodynamic biomarkers that would be supportive of moving therapy projects all the way into the clinic, rather than them falling short. And I think we need to have those kinds of programs that essentially, sort of the tide that lifts all boats in place. And the hope is that as we sort of think about the cooperative program writ large in the next year, because we are thinking about sort of reissuing those program announcements of significant changes, we could imagine having those aspects included in those particular areas. I mean, for me, the particular focus that I've taken on is biomarkers, and the other piece is mechanism of action. I think you've highlighted a couple of other ones in terms of databases and infrastructure, etc. Some of those things are also being dealt with at other places at the NIH, sort of in terms of the big data consortium and other places that we need to engage further in. I see Walter has a hand up to maybe address the question.
So it's a very common question, and it's a little bit thorny issue. Clearly, to make progress in developing new therapies, you need to have a lot of background work with regard to natural history of the disease, because that's what you're trying to change. You need some evidence of biomarkers that the therapy is actually doing what you think it's doing. So, as Rajesh mentioned, I think there's a new interest on our part to develop the biomarkers. The NeuroNext program is also a multi-site network to develop new therapies that are quote unquote biomarker informed, so actual defined studies that are very close to establishing something that would be important for a therapeutic trial, that those pre-trial studies could be done in NeuroNext. So for instance, the first study to be done in NeuroNext is an SMA biomarker project to develop a biomarker that could then go into the clinical trial. Those are fairly advanced situations. A lot of the research that gets you to the biomarker comes earlier in R01 research, and maybe with Rajesh's new program. Right. Well, then, so then that's the thorny issue. The thorny issue is the biorepositories and registries, because we have some say 600 diseases. Every disease would need a registry and a biosample. We haven't quite figured out how to do that without breaking the bank. Yeah, right. That would be great. Yeah. So we're doing that with Parkinson's, for instance, now. We're partnering with the Parkinson's organizations to build a biorepository and a database for that kind of stuff. Right.
Okay. So there were other hands up. So if you could put your hands up again. Okay.
Thank you. You mentioned that new applicants for these NIH grants are maybe at a disadvantage to people that have done it before. Is there a resource for somebody that's going to apply for their first one that gives exactly how these things are scored, so that we can put in the appropriate information that you're looking for, instead of giving you information that's not helpful for you making decisions?
So I think there's a variety of different resources, and I know Kip's going to jump in here as well. And it really depends. So first, the NIH, as far as the SBIR program, the NIH does have a workshop, a yearly workshop. This is NIH-wide, and it has all sorts of information. We provide different sessions on all components of the grant, everything from how to write a successful grant to how to actually do the budget pages to how to submit electronically correctly. It's usually about a 2 to 3 day workshop, so that's a great place for new companies who are interested in the programs to go. In addition, they can always contact the program director, or you have them contact me. I mean, part of what I do is help companies through the process. NIH is not a black box. There is a system here, and I'm happy to discuss the entire system, talk about the entire process, and help talk about their specific application and some of the pitfalls that they may have, because each scientific area, often there may be specific things that they should be watching out for, they should look at, they should make sure that they mention in their application.
Yeah, I'd just like to echo that the program director is probably one of the best resources. We show up to all the reviews in our scientific area. We see similar sorts of applications go through, what does well and what doesn't. And honestly, half my time is spent addressing exactly what you're talking about, talking with people who are new or people who are even very familiar but writing an application that's a little bit different than what they've done before, and helping advise them as to, we can't write a grant for you, but we can at least give a second voice to what we've seen in review previously and how they may respond to it.
And I'd like to just make one last quick addition. I think when companies work with advocacy groups, they tend to write better applications, not necessarily because they now know all about the grant process, but because they're able to write an application that's targeted to a specific market. For small businesses, that's really important. You need to know your market. You need to understand the need that you're trying to address and be trying to address a real need. Where some companies fall down is that they have a tool, but they're trying to figure out where to apply that tool. They tend not to do as well, versus something that is really targeted and tailored to a specific need in a specific market. Those tend to do better.
And one more thing, reviewers are very clued in now to the difference between statistically significant difference and functionally significant difference. And having advocacy groups helping them define what is meaningful to the patient is becoming more and more critical to doing well in review.
I've been listening this whole time trying to figure out how to formulate this question, so I'm going to put it out there and maybe you'll help me. So early on, I was talking to a scientist who was very big in proteomics, and they were looking at basically looking for biomarkers for for the condition I represent, for hydrocephalus. But they basically, in anything where they tried to submit for an R01 grant, were told, it's basically a fishing expedition, and it's low probability of getting funded. I was surprised to find out from him recently that he's working through the SBIR process to actually do the same research. And I was wondering, what is it that is different about the review process that would allow someone to be doing a fishing expedition in hunt for valuable biomarkers for hydrocephalus that would score, or maybe it's the bar issue with the funding level that might allow them to be successfully doing that?
So there's a variety of different things. So the SBIR mechanisms are fundamentally different from the R01. So the focus of those mechanisms are different. In addition, the way the program is structured is very different. And I'm going to try to kind of speak in general terms. So the small business program is set up into 2 phases. These are different phases from a clinical trial phase. The nomenclature does get confusing, and my apologies. So in the phase 1, SBIR is actually much more similar to an ARC-21 than an R01. It's a much smaller application. It doesn't have to have data. So you do not need preliminary data to get a phase 1 SBIR, and it's supposed to be basically a feasibility study. hey, let's see if you get something. And if you do get something and you are successful, then you can move to a phase 2, which is a much larger reward. And if you want to look at an equivalent, maybe you can think of it as an RO1. It's hard to speak in terms of RO1s and R21s, and because the focus of the SBIR program is so different. While an RO1 and an R21 is a very hypothesis-driven grant application, and SBIR is really to be driven towards a product. So there's a couple reasons why that could be the case, and it's probably that they were much more focused on the product at the end of the day and doing that feasibility study.
So generally that way of looking at things that might otherwise be lacking at the hypothesis but would have great value if you can couch them in those terms credibly, that's a very valid way to pursue funding of something that otherwise might be viewed as fishing.
Yeah. So I think we have time for maybe one more question. That usually kills the questions, which is why I usually say that. Okay. All right, Roger.
Well, Jess, one of the problems that we see at the Epilepsy Therapy Project are people who have a very early project, which is not really our thing, but they not only do they have a very early project, they really haven't a clue. They don't know the difference between a molecular patent and a use patent. They are misled in terms of how fast they think they can get the drug from where they are to the next step. It's a relatively common problem, and we spend a lot of time trying to help them. Do you have a contact point, like yourself, since you know how to do this? Do you have a contact point in the Institute that we could send people like this who have done actually some really nice laboratory work, but the next step is going to be difficult? The path is not clear. They don't have a path, etc. I don't think I need to describe it anymore. I think you know this picture.
No, I think our job is to try to help these folks move forward. So in that sense, I think sending them our way is the appropriate thing to do. I think it's a partnership between people in my office who sort of have specialization in sort of thinking about translation, but also then partnering with those program directors in the extramural division that really have the disease expertise. So in this particular case, those program directors who are epilepsy experts who know the vistas of research that are out there to really guide these newcomers to figure out how to navigate the road and what they might need to do. So we are forging those partnerships internally to strengthen our interactions so that we can speak with a common voice when somebody comes in through the door. So in principle, yes, I think you should be sending them our way, and we should be trying to help them.